So I'm Jennifer Cromley, and I'm presenting on behalf of myself and Nora Newcomb on using diagrams and visualizations to support science learning. So in this talk, we're moving a little more towards the classroom. In the practice of science and in science textbooks, these visual representations are ubiquitous. And when we talk about visualizations, we mean the full scope of diagrams, photographs, graphs, maps, all of those things that are not just words on the page. Folks who have done work in coding these representations in textbooks find that whether you're looking at middle school, high school, undergraduate, there is about one representation per page. And this is something of a change if you think back about 20 to 15 years ago. There was really a change in publishing, and it became, the technology of publishing made it much easier to just put a visual on the page whenever you wanted it. So for those of you, I mean, I was in middle school in the 1970s, and it cost a lot of money to put a color picture in a textbook. And they were reserved for special little sections of the book, and there weren't very many of them. Now, it doesn't matter. It's all just, computer generated. And it's super easy to put a lot of visuals in there. And in fact, they're used by the textbook publishers to sell the textbooks to the districts. But as the other two presenters have said before, these visual representations convey really important content, especially spatial relations and functional relations. So they are not just pretty pictures on the page. But surprisingly, student comprehension of these representations is poor, and you have heard that from these folks as well. But there is just increasing and really accelerating body of evidence that students don't understand these visual representations. And that evidence comes from a lot of different kinds of research paradigms. You can do eye tracking, meaning looking at where students are looking on the page, and there's especially some recent work from Europe, looking even at pretty young groups, 5th grade through undergraduate, that what students are looking at on the page is not necessarily the important stuff that they need to be looking at. You can give students textbook pages or other, just freestanding diagrams with captions and labels and such, and have them say what they're thinking while they are trying to make sense out of that. And you will see that they do not say very much about the diagrams, and when they do, they often misunderstand what's in the diagrams. You can give them questionnaires about how much they feel that they learn from diagrams. And again, this is some recent European work, and they'll tell you honestly, I don't get much out of them. And another interesting way of finding evidence about comprehension of these visuals is to say, okay, we have national and international assessments like NAEP, PISA, TIMSS. These have visual representations on them, and you can compare the difficulty levels of items that have a visual and items that have no visual, and you see that there is this difference in the difficulty levels on those items. So you might be thinking, oh, come on, these kids are very visually literate, they just look at the picture, they know what it means. But here is an example from one of the visuals that we created teacher supports for students to understand from one of the units that we modified in the center. So this is from a traditional curriculum, from a Holt curriculum, and it's a book called Sales, Heredity, and Classification. It would be covered, this unit would be covered in about 1/3 of a year in a typical middle school classroom in a book that's about 125 to 150 pages long. And there is an awful lot going on in this diagram. And I'm going to show it again after I talk a little bit about some of the difficulties that we know from cognitive science research. But keep that in mind, because I think that might not be what you imagine when you think about a picture in a middle school science textbook. So what are the difficulties? And these are all grounded in the research. But students can ignore the visuals or spend very little time looking at them. Now, why might that be? They could have a false sense of comprehension. Their eyes sort of skim quickly across the picture, and they sort of have this false sense that they have comprehended it. Some students tell us, well, I didn't spend much time on the diagrams because I know they're hard. It would take a lot of work, and I just don't feel like it. Sometimes they are honest with us about that. And to some extent, and Dr. Shai didn't talk much about this, but there is a whole schema for how to understand these things, and they may lack a schema for how to make sense out of the diagrams. We also know that there's a role of background knowledge, which I think both of our presenters alluded to, that the more you know about these topics, just like in text comprehension, plays a really important role in whether you can make sense out of that diagram. And it's not just that the diagram replicates exactly what you knew before you looked at it, it's that you are bringing that knowledge to bear, and as both of these presenters talked about, you actually need to make inferences. So those could be local inferences, sort of within the diagram itself or between a reading passage and the visual, or it could be elaborations from your prior knowledge. So you might be looking at a diagram of the heart and you bring to bear your knowledge about how every cell in your body needs oxygen, and the main purpose of the heart is to, well, one main purpose of the heart is to move the blood out to the lungs to pick up the oxygen, drop off the waste carbon dioxide, come back to the heart, and get pumped out to the body. So you might wonder why. How come you can't just look at that picture and get it just by looking at it? And I think that the answer is, if you think about reading, that's a relatively recent cultural invention. You could go back to about 3000 BC, and that's about when human beings started writing things down. And once you start writing things down, of course, other people need to know how to read. If you think about our Arabic numeral system, there's some debate about the dates, but somewhere between 200 and 800 AD, people started using this numeral system. Of course, we have to teach kids how to read because it's a recent cultural invention. Of course, we have to teach kids how to do math because it's a recent cultural invention. There is, again, some debate about when these representations came to be, but a lot of these canonical representations are from the Enlightenment. They are very, very recent cultural inventions. So thinking that a kid can look at these and get it really ignores the fact that human beings invented these ways of representing something out in the world, microscopic, macroscopic, or at human scale, and that for many, many kids, we need to teach them how to do this. There are a bunch of promising classroom inventions that are relatively recent. I'm citing myself and my team a lot here because not many people have gone into classrooms and tried this out. But what we did in the center is what we call conventions of diagrams, which means we just explain to them, this is a caption. You should read the caption. It's the main idea of the diagram. But we also teach them about arrows and scaling and color coding and different kinds of labels that name objects or that explain parts of the process. We've done work in self-explanation, building on Dr. Chi's work, self-explanation in diagrams. We've given students partial diagrams and asked them to complete them, and we've done that on pencil and paper, and we've also done it with artificial intelligence software called CogSketch. We have had them just read the passage, and when they get to that thing that says see figure 2, they actually draw an arrow over to the figure, but they also do things like look back into the text if there is a label that describes something and they don't know what it is. So trying to get them to go back and forth between the text and the diagrams. And we've also done work on space retrieval, which you're going to hear about later, and we have some of the national experts in the field here, but we've done that using diagrams, and many of my collaborators in the room are in the room, so I won't name specific names. But we know that we can take these research-based principles from cognitive psychology into the classroom, and we get some pretty good effect sizes. So this is just one recent manuscript that we're working on with conventions of diagrams and coordinating multiple representations. We replicated it in 2 schools, and these are effect sizes pre to post test on the left, so you can see In some studies, we're getting effect sizes of about 0.5, and in some of them, about 0.4. We're post-testing kids on biology knowledge, on biology diagram comprehension, and looking for transfer to geology diagrams, which we didn't get in some of our work, but actually with this coordinating multiple representations, we actually got a pretty healthy effect size on this. So this is just one example, but that's the kind of bump we've been able to get. I wanted to show you a little bit what some of these look like. So in some cases, we're just teaching students, as we did in the center work, here's a label saying this is the pollen. There's an arrow taking it of the white flower to the stigma of the purple flower. Then there's a caption that explains the main idea of the figure. There's also some text that goes along with it, and it explains about Gregor Mendel, and it says, as shown in figure 1. So sometimes we have them go back and forth here. And in the self-explanation, we found this little confused guinea pig. He gave bad explanations. And Bradley Berge, raise your hand, Bradley. Bradley worked on this intervention. So the confused guinea pig gives a bad explanation, and then or expresses a confusion, and the students have to give a better explanation. So in terms of real kids working with real materials, that's sort of what that looks like. So in the center, just to tie this into the rest of the day, our idea was to build basic skills in comprehension of these visualizations, those conventions that are unique to the visual representations and that you don't find in running text. Captions, labels, arrows, cutaways could be a cross-section, or it could be actually sometimes cutting out a quarter of an object. Color coding, enlargements, which in some ways tap into this issue of scale and scaling, which I believe Christine Massey is going to talk about a little bit. And then we did do some work with table, columns, and rows. There are not many graphs per se in these textbooks, so we didn't do a whole lot of work on that. And we are not assuming that understanding these conventions is automatically going to lead to some fantastic spatial or other inferences. We're really thinking of it as a necessary prerequisite for comprehension of the visuals. So to come back to this example, we have labels that name objects: mitochondrion, chloroplast, light energy, ATP. We have these arrows that are connecting in this case, it's actually an animal mitochondrion, which is releasing carbon dioxide and water, which goes over to a plant chloroplast. There's, it's a little hard to see on the slide, but there's this enlargement from the plant cell. It's showing that this is the enlarged chloroplast. So you have to think about scaling. Kids can't really see cells. Sometimes they do the cheek scrape and look at it under a microscope, but you definitely can't see the chloroplast, not without a scanning electron microscope. We have cutaways. The plant cell is cut, the animal cell is cut, the mitochondrion is cut, and the chloroplast is cut. We have this color coding. Light energy is coming in and it's orange. ATP is capturing the energy released from the mitochondrion, and that's shown in orange. There's a caption up here that's shown in purple, which accompanies this purple arrow, and an explanatory label about photosynthesis, which is accompanying this red arrow, which shows glucose and oxygen being released, actually being captured in the chloroplast, which is then eaten by the animal over here. There is an interesting misconception that this particular graphic might unfortunately reinforce, which is that the mitochondria are not just in the animal cells over in the right side here. They're also in the plant cells here, because the plant cells have to metabolize that glucose that they have created. So that's another thing that we worked on, and again, Christine Massey is going to speak to this. So I'm going to go a little overtime here, but how did we instantiate this? The request for proposals asked for modifications to existing curricula. We were asked not to reinvent the wheel, not to create new curricula, but to take these existing curricula that are widely used in the schools and to modify them using certain specific principles of cognitive psychology. So what we did was instructed students in these conventions of diagrams, and the science teachers would give this instruction verbally at strategic moments during their regular instruction. We do have two teachers who were involved in pilot work who are here today, which is great, so you might want to chat with them at the poster session or over lunch. They would follow the district pacing guide. So whatever the district said, you are teaching on Tuesday of the following week, they would follow the pacing guide, but when they sort of reached that point in the textbook, when there was a visualization, they would stop. They might give a brief explanation, they might ask students to complete an exercise, the students might, or the teacher might identify conventions such as naming labels or captions, they might create their own captions or draw in arrows that we had taken out, or create their own diagram, or give suggestions for how to improve a diagram. And then there was some whole class discussion. So there was some small group work and some large group work. And somehow, I've lost my, all right, well, so that's the picture of what we did in trying to take these laboratory principles of cognitive psychology, mostly developed from understandings of European college students as they looked at diagrams, and put them into classrooms with teachers in a national center in 180 schools. So I think we should now try to get a synergistic conversation going. But thank you for your attention.